Okay, perfect. Hi, my name is Emily Chen, and my thesis is on the application of environmental DNA for monitoring and management of aquatic biological invasions, emerging trends and advancements towards best practice. So the global spread of aquatic invasive species, or AIS, poses serious threats by jeopardizing ecosystem services and impacting sustainable economic growth. Human-mediated pathways and vectors, such as intentional trade of aquarium species, ballast water from international trading, and aquaculture, have overcome biogeographical barriers that would have previously prevented species from successful invasion. Once AIS establish themselves as part of a new ecosystem, it's nearly impossible to eradicate because of their rapid ability to rapidly grow their population, which ultimately leads to widespread invasion. AIS can devastate native habitat balance by outcompeting indigenous species for food, restructuring food webs, and introducing harmful parasites. And apart from ecological damage, AIS are largely linked to economic losses through causes such as increasing food production and labor costs. So until recently, the majority of studies focusing on biosecurity have conducted surveys such as plankton sampling and settlement plates with subsequent identification using microscopy or visual observations of species. However, this can be time-consuming and dependent on specialized taxonomic expertise. And these studies, depending on species identification, can be challenging and lead to bias, especially when dealing with larval or juvenile stages of many taxa. So more rapid approaches, such as visual rapid assessment surveys, can be conducted at easily accessible sites, such as marinas, but these surveys also do rely on experts in multiple taxonomic groups. There's also the high risk of misidentification, particularly if it is the first sighting of a species, because prematurely assigning a new name results in multiple names globally for the same species, which can cause confusion, as seen in the publication of Stefaniak et al. 2009. And the gaps in these traditional surveying methods are pushing researchers to rapidly develop novel methods for use in surveillance and monitoring programs. And an emerging approach that has proven to be effective is the use of environmental DNA. So environmental DNA, or eDNA, is DNA basically released from organisms into the environment in the form of shed cells, feces, gametes, etc. And it's collected from samples such as soil, air, and water. And depending on the goals of the project, researchers are actually able to utilize assays to target specific species of interest or use a community-wide approach through metabarcoding. This is a relatively new field with an exponential rise in eDNA research since 2008, as seen here. However, this method has already been implemented for many applications, including but not limited to dietary assessment, invasive species surveillance, and measuring biodiversity patterns. And some advantages over traditional surveys are that the sample collection is a non-invasive process, and it's a rapid and cost-effective way to infer the presence of a new potential AIS so that management can then be informed about where to conduct more targeted surveys. So the main objectives of this thesis are to recognize any patterns in taxonomic groups and geographic regions, seeing how marine and freshwater studies differ in methodologies, investigating the usage trends for different laboratory protocols, and providing recommendations based on the gaps in the available literature. And collating data from current literature to recognize patterns of AIS detection in eDNA studies will be useful for informing future research efforts. So to find literature that use environmental DNA to target AIS, keyword searches were conducted on the following databases in step 1. And articles were included if they were published from January 2010 to April 10th, 2020, which was when the literature search ended. And after screening, it's important to note that studies that used multiple methods were inputted as distinct records originating from the same reference. There were 11 variables that were extracted for both species-specific and metabarcoding approaches, and a total of 492 records from 192 unique studies were ultimately used for analysis. And this flowchart just shows some of the technical variables that were extracted from sample collection to the DNA extraction method and to the different analyzing processes for both species-specific and metabarcoding approach. And for the following section, the results and discussion will be combined for each group of variables to present a more logical flow. So although there has been an exponential growth in eDNA studies overall throughout the last decade, as we've seen before, the temporal trend of using eDNA specifically for AIS detection is not as clearly defined. However, it is very clear that there are more freshwater than marine records and more species-specific than metabarcoding. There were 28 total countries that met the selection criteria in the methods, and this kind of clustering in North America, Europe, and Australasia is consistent with previous analysis on the geographic distribution of general invasive research. However, these gaps in South America and Africa don't signify that there aren't working groups using DNA biomonitoring. In fact, many studies that were excluded during the screening took tissue samples of AIS, used eDNA in terrestrial ecosystems, etc. So it's not surprising to see that the eDNA studies of AIS were dominated by freshwater records due to the dynamic environment in marine systems, including increased mixing, higher levels of dilution, and physical processes such as upwelling. And despite the potential of using eDNA in marine systems, there are many considerations that need to be taken for successful large-scale implementation and reliable ecological inferences. And across the 10-year period, studies have mainly focused on the targeted detection of single species as opposed to multi-species community metabarcoding approach. One explanation could be that there have been recent comparison studies on the sensitivity of approaches, with the overall consensus being that when specific target species are known, a targeted eDNA approach would ensure higher precision and be a better allocation of resources for AIS management goals. And most eDNA studies targeting AIS are concentrated in Europe, North America, and Australasia, as we've seen. But because there are aquatic ecosystems that are ecologically and economically important in these understudied regions, they should be the focus for future invasion research. So to determine the pattern of taxonomic groups studied globally, the percentage of studies done per taxa for freshwater and marine environments on each continent were calculated. And for freshwater studies, besides South America, which only had 2 studies done on freshwater species, the most studied phylum on each continent was Chordata, ranging from 34.1% in Europe to 79.3% in Australasia. And besides Chordata, there is not a clear trend of the percent of targeted species. And for marine environments, there is actually a lesser number of phyla studied on every continent compared to their freshwater counterparts. And with regards to between taxa, there's also not a very clear trend. There could be less taxonomic diversity of studies in South America and Asia because of the limitations in financial resources that directly correlate to research efforts in those regions. So even if there are concerns of invasion, studies that target those species and actually do find presence may not be able to further investigate if AIS management is not given appropriate economic resources. And the main group studied in freshwater environments in this analysis were chordates. And these observations could probably be explained because commercially important activities such as fish farms need to be monitored in case non-indigenously farmed species escape to wild ecosystems. However, the study of invertebrates comprise a majority of marine studies, and this supports the findings of Bellard et al. 2018 that revealed a taxonomic bias in invasive research towards invertebrates. And this may be because some of these invasive species are causing significant ecological and economic damage. So basically, if invasive invertebrates have the greatest impacts in marine environments, they will correspondingly be the most thoroughly researched. So this figure just shows the type of eDNA samples collected from metabarcoding, and it is very clear that samples taken from some point in the water column dominated over any other type of collection method, with ballast water only having 7 records using that as their sample source. And for these figures, it shows the usage in % of different extraction methods for both species-specific and metabarcoding approaches. And there is really no one single common method used, as we've seen, but the CTAB extraction method, which is the pink bar, its use is actually relatively consistent for a species-specific approach from 2013 to 2019. However, in the same time frame, this method is decreasing in use for metabarcoding. And there were only the 3 main extraction methods were directly compared. There were actually 18 other methods that were placed in the other category, and these were mostly commercial kits, but also study-specific extraction methods. So with regards to the change in amplification technique, qPCR, which is the purple bar, has consistently increased in use from 10 records in 2013 to 60 records in 2018. And qPCR actually became the dominant PCR method in 2016 to currently now in 2020. And digital droplet PCR, or ddPCR, which is the pink bar, started to come into play in 2015. So because most studies don't provide justification for the choice of eDNA extraction methods, the use may be simply related to requirements and existing equipment in each laboratory. This could also explain why there were many instances in which certain working groups had consistently used the same extraction method, despite targeting different taxonomic groups. After successful extraction, there is a number of different amplifications that can be applied for species-specific studies. And the shift in the method of choice from endpoint PCR to qPCR is due to its increased sensitivity, more quantitative measurements, and faster processing times. However, new developments are continuously being produced as eDNA research advances. In our analyses, digital droplet PCR came into the picture in 2015, which can detect species at low eDNA concentration and appears to be less affected by enzymatic inhibition. This technique may not be widely used in detecting AIS yet, potentially due to the cost and lack of equipment availability in laboratories, since it's still in its early stages. So for these four phyla, the CO1 is the most targeted gene region, with exclusive use on the 10 records on polychaetes on the bottom right corner. And on the bottom left, mollusks were actually the only group of studies that targeted more than 2 gene regions, but CO1 was still the dominant region used. And CO1 could be the most targeted gene simply because of the higher level of coverage in genetic databases. In fact, GeneBank has seen a significant increase in CO1 records of sequences from 8,000 in 2007 to roughly 2.5 million in 2017. However, previous studies have shown that CO1 may not be suitable for detecting some taxa and for use in metabarcoding. And consequently, there have been recent country-specific barcoding campaigns, and this will help design primers for species present in those specific geographic regions. So certain metrics were excluded from analysis because they were not directly comparable between species-specific and metabarcoding approaches. For instance, the eDNA sample type was presented for metabarcoding approaches, but not for species-specific, because there were many studies that were not explicit enough in their methods to confidently determine where in the water column the samples were taken. There was also the time constraint that the literature search only took place over roughly 1 month, so relevant studies may have been missed. And also, only fully aquatic species were included, and they had to have been invasive to meet the selection criteria. And another consideration is actually the relative weight assigned to each study, because some references had a higher effect on the results than others, such as Roy et al. 2018, which was responsible for 13 records in that year. However, choosing to input each methodological variation within a study as separate records originating from the same reference was more appropriate for analysis, but it's something that needs to be taken into consideration when making conclusions based on the results. So we recommend that there should be a common workflow diagram or decision tree for each stage in the experimental design, and previous studies have proposed small-scale versions of this, such as Evans and Lamberti 2018. However, having a highly respected and international network, such as DNAquanet, providing a basic foundational common diagram that researchers can then tailor for their to meet their research objectives will be beneficial for future eDNA studies.
2 more minutes.
There should also be an improvement in reporting guidelines, such as having a minimum set of criteria for metadata collection, and this will help increase comparability between studies. And terminology also needs to be standardized because bulk tissue DNA and eDNA are often used interchangeably for different sample types, and the phases of invasion aren't consistent across the literature. This leads to confusion for researchers when comparing studies, as well as for policymakers when eDNA results are presented to them. So in conclusion, because we've seen such a high variation in different sample processing and laboratory protocols, and there were some variables that didn't have a clear trend, there needs to be a coordinated effort for implementing some common guidelines. We recommend having a standardized set of criteria for data collection, as well as a common diagram outlining the best practices during each stage of the experimental design. And because only descriptive statistics were taken out and this thesis is mostly qualitative, we advise the readers to take this analysis just for use in identifying general trends. And overall, future refinement of methodologies and a greater understanding of eDNA ecology will improve the quality and reliability of future eDNA research. I want to thank my supervisor, Luca Mirman, as well as the Marine and Freshwater Research Center and GMIT for hosting me. And thank you all for listening.